Hello, everyone. Welcome and thank you for joining today's webinar on the topic of the power of soil biology data in guiding farm practices. I wanted to extend a special welcome to our BeCrop advisors. These valued advisors are an extension of Biomaker's team who have full access to our educational programs, tools, and technology. They're helping farmers across the globe save money and recover their soil health by applying the right products in the right places based on their soil needs through the power of BeCrop Technology. We started this free certification program over a year ago, and today we're proud to announce that we have over 800 registered in the program across the globe. I will be back at the end of this webinar to provide further details about the BCA advisory program, and we will also follow up with an email in case you're interested in joining. Before we get started, I would like to go over some housekeeping items with you guys. Please use the question pane and add all your questions throughout the webinar. You don't have to wait until the end. We have dedicated time to ensure that we answer all your questions. If we don't get to them and we run out of time, we'll make sure Dr. Purnell or one of our team members gets back to you with an answer. All of our webinars, including this one, is recorded and available online. A link to the recording of the webinar will be emailed to you within the next 24 hours. But you can also visit us on our website, go on to our resources section, and check out all of our webinars that we have done throughout the last couple of years. Last but not least, we have released a new tool for our BeCrop advisors, which is a project planner. This is a tool that helps create projects easier in minutes with smart calculations around numbers and samples. If you're interested in this portal, we'll also be sharing an email with you at the end or within the next 24 hours, and you can get in touch with us and we'll be more than happy to provide a demo. And now, without further ado, I would like to welcome today's speaker, Dr. Jacob Parnell. He's a soil microbial ecologist at Biome. Dr. Parnell works with farmers, researchers, and producers to examine the impact of management practices and agricultural inputs on soil microbial community. Dr. Parnell has led projects discovering and bringing microbial products to market and has extensive experience in designing large-scale strategies for understanding the impacts of climate change and agricultural practices on soil health. Dr. Parnell received his PhD from Michigan State University and did postdoctoral research at Utah State University prior to joining the faculty as a research assistant professor. In 2011, Dr. Parnell accepted a position as a microbial ecology staff scientist for the National Ecology Observatory Network. From 2014 to 2021, he worked as a staff scientist with Novozymes in their microbial biology group. So Dr. Parnell, thank you so much for taking time and being a part of our webinar today. We will be exploring a case study that brings to light the crucial insights provided by BeCrop Technology used to develop effective strategies for promoting soil health with a special focus on minimizing diseases with early risk detection. I will now turn off my camera and I'll be back after your presentation to get to the Q&A and talk a little bit more about BeCrop Advisory Program.
Thank you, Sarah. I appreciate the introduction and the opportunity to share some of this information. We have a pretty cool study to share today. I wanted to start off with kind of giving an overview of what I'm hoping to discuss today. One of the things that we're facing, I think we're at an inflection point where kind of the time is right. We've got all of the policies are set in place. There's a lot of consumer information about soil health, and so it's a time where making some more regenerative approaches to farming is quite palatable, and it's something that we can start to look at. And so how do we kind of maximize some of these changes? And then I wanted to kind of look at a case study that we have where we've actually kind of looked at, in this case, we're going to look at a conversion from conventional tilling to no-till and how that impacts the soil health, how that impacts the biology of the system, and how that also, how there's kind of a feedback from the biology and the crop. There's kind of an interplay between those. And then finally, we'll go over a tool that it's not necessarily a new tool, but it's one that is, I think, a little bit undervalued. That is one that we could really start to look at a little bit more and explore the value of this tool, which is the BeCrop Rate. So really, right now, you know, this inflection point that I was talking about, we've got an increase in synthetic fertilizers. Not only that, but there's also kind of this almost like this public distaste for synthetic fertilizers. People want to go more natural, regenerative, in soil health, sustainable agriculture. So that on one side, we've got kind of this headwinds of economics against the synthetic fertilizers, and we've also got this, you know, this momentum behind the community that is asking for this change. And so the question becomes, you know, how do you know what practices or what products are, you know, the right ones to use? So on one hand, we have this kind of big change that's going on. On the other hand, we need to produce more and higher quality foods on the land that we've got because of increasing population. We've also got a lot of countries that are moving up kind of in this socioeconomic status. And so kind of the demands of food and the quality of that food are increasing. So we kind of have these two different storms that are building that are and pressures on agriculture. And so we really want to look and see how we can kind of make a difference there. And so that's kind of the I guess the spark that helped build Biomakers. Biomakers was first founded in 2015, and really what we try to do is we try to look at the biology of the system. And so we really see the microbes as kind of a bioindicator of what's going on in the soils. There are billions of microorganisms per gram, thousands of species within that, and each one of those species has you know, likes and dislikes, or you know, a preferred temperature, preferred carbon source, nitrogen source, all of these things. And if you change any of those, then you're going to change the whole system of that biology. You're going to, you know, you're going to benefit some at the cost of others, and so populations are going to shift. And so by looking at that biodiversity, we can get an insight as to what's going on in the community, how it's functioning, and how it's providing a healthy environment for crops. And so we see it as kind of an ecosystem, what we can look at to see how we can improve yield, improve food quality, and also we can look at what products, what management practices are best for that system. So I'd like to introduce our BeCrop technology. This is kind of how we're addressing that, how we're using biology as an indicator for basically anything that's going on in the soil. So what we do is we will collect a soil sample, and we will sequence the DNA in that soil sample. From that, we're able to pull out of that you know, who is there, who is in that soil sample. So, you know, if you've done some kind of a new practice, or if you just want to say, I want to see the status of my soil right now, go collect a sample, you send it, we sequence the DNA, and we say, well, these are the organisms that are thriving right now, these are the organisms that are a little bit depleted, and these are the functions that they carry out, and we put it all into a big report that is easy to digest, easy to interpret and understand. And so within that, we're able to look at kind of the variation in the community. We're able to look at carbon cycle and whether you're the processes in your soil are there that are sequestering carbon or whether you're releasing carbon from the soils. We identify different bacterial pathways that are involved with nutrient cycling, health, you know, some microbial interplay between the plant health and abiotic and biotic stresses. And we also deliver it in a very intuitive way so that anybody that looks at this can guide it, get an idea and say, okay, I understand what's going on with this report. So we're not going to give you, you know, a giant many megabits file full of tables and figures. It's going to be easy to understand and easy to be able to figure out what you might need to do or what you might advise these growers to be able to do in their fields. And so this BeCrop technology, we've kind of focused on three aspects that we can talk about today. One is just a simple test. This usually is something that a farmer or an advisor would apply. And really what this does is it's a way of saying, what is the status of my soil right now? So what does my soil look like? What is it lacking? Where is it, you know, functionally, how is it beneficial? Kind of gives you a status of the soil. Or you can actually look at several different ones and say, you know, I've got underperforming yield over in this area relative to this other area. And so I'm going to collect samples here and here and see what the biology is telling me. And we're able to kind of look and say, well, the nutrient cycling of this area, it might be different, or you might have more disease pressure in that area that's not manifesting in, you know, full-blown disease breakout, but it is kind of, I guess, lowering your yield. So we're able to look at some of those, and this would be kind of at a grower level. The next level is BeCrop trial. So using the same underlying technology, we're basically able to do the same approach and say, you know, if I've got a biological product that I think is the next best thing, what I want to do is I want to take it out and I want to test it in the field. I want to see if it actually works in a grower's field. And so we're able to say, well, you've got it, you're untreated versus you're treated, you're going to have it replicated, and we're going to be able to tell you this is how your biological product or your management practice or whatever it is, this is how it's impacting the biology of that community relative to that control. And then this third area is this BeCrop Rate. One of the things that we've found is we've done more and more of these samples. We've sampled over, we've got over 40,000 samples that we've analyzed. And as we build this database, we can find more and more patterns within that database. And one of the patterns that we found is that we're actually able to, based on some of these microbial indicators, these bioindicators, we're able to tell you kind of almost like a grade of how healthy, how regenerative and biosustainable your soil is. And so if you're using conventional practices, synthetic fertilizers, it comes back as a low score. If you're using regenerative practices, then it comes back as a higher score, and almost like a grade. And so this is the way that we can kind of say, you know, people claim that they're doing regenerative agriculture and that they're building soil health, but are they really doing it? Is it really showing up in the biology? So these are kind of some of these pillars that sit on this platform of our BeCrop technology. And they come out in nice, easy to identify reports. Here's an example of how it would report. You would have your samples, you would have different functional categories. And then you would be able to say, you know, for example, yellow is low and blue is high. And so you'd be able to say, well, you know, I can see which ones have high functional capabilities and which ones have low functional capabilities, or, you know, these soil health indices, nutritional pathways, disease pressure, whatever it is you're looking at, you'll be able to see it in an easy to read format like this. And so there's also a user-friendly report that comes out that gives you a lot of the information that people are looking for in traditional soil biology tests, fungal to bacteria type of ratios, actinomycetes to ectomycorrhizal fungi. And so it just gives you a nice, easy to read, intuitive way of understanding your soil. All you would need to do is you would order a BeCrop test. We've got an online portal now where you can go and you can actually do this all online. And so you can just order one of these BeCrop tests. We will send you a sampling tube. All you do is collect the samples, write down the metadata. So you make sure to say, you know, is this an area of low yield versus high yield that I want to compare? Or even tell us, I want this sample to be named whatever it is. And then you send that in to us. We do all of the work, and within probably about 6 weeks, we're able to get you back this full report where we're able to say, this is what we saw in your soil samples. This is what your farm looks like, the biology of your soils. And so I'd like to kind of look over a really quick case study that we did that I thought was pretty interesting. And so this is one that I wanted to share with you. This is actually, we have a program called Fields Forever where we work with to look at groups that are working on building soil health. And so this case is one where what we actually looked at, this was conventional till, and then they compared it with no-till, and they wanted to look at the microbiome. And this is with a wheat crop. And so we had 3 locations that were no-till. We had 3 locations that were conventional till. And it wasn't until later that we actually found that we had a different variety of wheat, which will play an important part, because we found out that this different variety of wheat that with the microbiome is responding differently to these conditions. And so we were able to tell that there was something weird going on here. We went back to our collaborator that was involved with this, and they said, oh yes, those fields had a different variety. And so we were able to actually pull that out. So we're able to find differences in just the variety of wheat in the soil biology. But what they did is they did 3 side-by-side comparisons. So that means that this Oberg field was close to this Essen field. The Port field was next to this other field, and so they had one field right next to the other one. And so most of the conditions they're kind of considering would be fairly similar between them. Like I said, we had 2 different varieties, and then they looked at 5 different time points throughout this crop cycle, the life cycle of the crop. And so some of the big things that we saw was we saw this shift in the microbial community. And this was one of the first things that kind of got us interested in thinking that there was something going on, because you see, especially here for example, in time 2, this is a non-metric multidimensional scaling. I think this one is actually a principal component analysis. And so really the axes don't mean a whole lot. What we're really looking at is that the closer these points are together, the more similar they are. The further apart they are, you've got very different communities. And so what we see at the very beginning is this time 0 is actually before you've done any treatment. So this is before they went and they tilled up one field and left the other field. So this is kind of at the very beginning to establish a baseline. And you can see that everything is just mixed. There's no distinct patterns of anything. And as we start to kind of move along this time 1, we start to see these X's are kind of off a little bit different than the circles. And that continues where we've got here in this time point 2 especially, you can see that these X's for this, now the blue is treatment and the X is one of the varieties. So we've got one variety where when you till the field, it basically pushes that microbial community from this area, and it pushes it over into this area. So it's kind of a shift, kind of going vertically. And then when you do that with the other variety, it's a shift, but it goes the opposite direction. So this is where we started to say, something weird is going on here, and we can't quite figure it out. We later found out that, well, these are 2 different varieties of wheat. So in this variety of wheat, we're shifting the microbial community in this direction, and in the other variety, we're shifting it in the opposite direction. And it stays somewhat consistent throughout the rest of the season. You can see that the blue is always going to be on the outside of those. And you can see that the X is always at the top and the circles are always at the bottom. And so this is basically telling us two things. One is that the variety itself has an impact, but also we're seeing an impact on the biology due to the treatment, due to till versus no till. This other one is looking, so that first one was looking at the bacterial community. The second one is looking at the fungal community, and we're seeing somewhat the same thing where, especially here in this time too, where we can see that there is a little bit of a difference between the fungal community of these. It's not as strong an impact as what we saw with the bacterial community, but we do see a little bit of a difference there. And so that's just telling us overall, what are the impacts of your treatment, your time, and the variety on the microbial community. So now I wanted to go in a little bit more and show that this bacterial and this fungal diversity, and this time I'm looking at it for each variety. So this is just that first variety where we had the two samples. And so you can see, especially here in time 3 is where we see kind of that biggest difference, where you can see that all of the conventional are on one side and all of the treatment are on the other side for both fungal and for bacterial diversity. So this is where we're seeing that the treatment really is showing a pretty strong difference in that microbial community based on this beta diversity that we're looking at. And this is for both bacterial and for the fungal diversity. So what does that mean though? And so one of the things that we can do with our BeCrop technologies, we can drill down into not only just the taxonomy, but we can also drill down into the function. So first I'm going to talk a little bit about one of the taxonomic differences that we saw that I think is interesting. And then we can go into some of the functional differences that we saw. So nitrifiers are microorganisms that basically take and convert from ammonium to a nitrate. And so these are very good in that nitrate is the preferred nitrogen source for plants. And so in being able to make it soluble to the plants, they're able to take it up a lot more. On the other hand, if you have too much of this going on and the plants aren't able to take it up fast enough, then nitrate is also very soluble and leaches into the groundwater. And so it's very important, this ammonium oxidation or nitrification or that's happening. And so one of the things that we saw, this is looking at 3 different, basically of the dominant nitrifiers in the microbial community. And so here on the left, I've got conventional, and on the right, I've got no-till. So here we can see that conventional, we've got about right around 1.5 to 2% consistently of the microbial community is this Nitrosocomiscus. I know I said that wrong and I apologize, but it's this organism. But then when we've got that under no-till systems, it jumps up to around 3 to 4% consistently. So we've got this shift of this Nitrosocosmicus that's going on in the no-till. But then if we look at some of these others, so this was Oleophilus. We look at this other one, we see kind of the same thing where under no-till we've got this increase, but there's this other one, and I apologize, I don't know why that's so blurry, but we have a different one where again, this is the same genera of nitrifiers, and we see that it's actually higher in that conventional till than it is in the no-till. And so these are actually some of the individual samples that were collected and that relative abundance of each of those samples. So you can see that it actually gets quite high up into the teens, percentage-wise, 10% or so, And so these are some pretty big shifts in the microbial community just as nitrifiers. And so the question is, well, what does that do? What does that do functionally? And so one of the things that I want to point out, so this down at the bottom is actually these samples were sent in for nutrient analysis as well. So this is actually the nutrient analysis, the traditional nutrient analysis that you would get. And this is kind of the feedback that you would get from our tests, the BeCrop tests. So one of the things that this inorganic nitrogen release, that's really kind of that process that's allowing the nitrogen to be able to take the plant available form. So anything that's converting to nitrate and making it available to, so this could potentially include nitrogen fixation. It's just saying anything that is pushing the direction for it to be nitrate is going to be in this pathway. And anything that is pushing away from this, so denitrification, for example, is going to be in this consumption. So basically it's saying you're competing for that plant preferred nitrogen source through denitrification. You're pulling it away from nitrate so that it's not available to the plant anymore. So one of the things that we see here is we see that it's highest, in this comparison of this port and Littler, and that's this one right here. So where we see this highest difference in nitrogen pathway, and this nitrogen pathway, by the way, is basically kind of a summary of those two. So it's, as we look at basically kind of the process of this, we're saying this is going to be positive, beneficial for the plant. So we see that it's significant and it's beneficial for the plant. And in this case, we're looking at port compared to Littler. And so when you look down at this, we see basically we're telling the same story, but we're telling it from a pathway perspective. We're telling it from, this is what's going on at the microbial level, the biological level, and we're actually able to see that as confirmed in looking at the nutrients. So we can also do this with things that are not as easy to, I mean, they're easy to measure, but for example, organic matter, it often takes quite a while for you to be able to measure any appreciable changes in organic matter. In this case, we're able to do it, and we'll talk about that in just a minute. This again is that nutrient analysis, and these are our tests. Again, this is just like you would see in one of these BeCrop portal tests that would come out. In this case, we're looking at aerobic respiration and organic matter release. And I want to really focus on aerobic respiration first, because in this conventional, you'll look, now the darker the blue, the higher that level is. And then it goes kind of to this, it goes light blue, then this color, and then a light yellow, and then this dark yellow, it means it's very, very low. So what we're seeing here is that under the conventional, we're seeing a lot of this dark blue. And so in this case, we're comparing this Esser to Oldberg, and we're seeing that there's a lot more dark blue. So we would look at this just visually and say, well, it looks like there are a lot more microorganisms that have these aerobic respiration pathways in this conventional till than there is in Oldberg. And same thing with this Mosier versus Jensen. So there's a lot more of the dark blue. There's actually none of the dark blue in this one. So in one of them, we're seeing that this dark blue is much higher in the conventional till. Now, if we go to the other variety of wheat, we see it's flipped around. So in this variety, we're seeing that we've got a lot more aerobic respiration in this wheat than we do in the conventional. And then another thing that I kind of briefly wanted to point out is just kind of this organic matter release, especially in the second variety, where early on, so I neglected to mention that these are each the triplicates of each of the 5 different time points. So here you have time 0, time 1, 2, oh sorry, time 1, 2, 3, 4, and this would be time 5. So you can see here that as we get to later on, we're basically really losing a lot of that organic matter release. It's happening quite a bit early, and it really isn't much different between the two. But the difference comes later, where the organic matter release really kind of, those organisms kind of disappear, where it doesn't in this conventional. And so here we're seeing a lot more organic matter release in the long run, and here we're seeing it kind of shut down. So when we look at these values, so for example, here we're seeing that in Esser and Oberg, which are, oh sorry, Esser and Oberg, which are the two that we're comparing, we're able to look at those. We're able to see how this is how it's happening and how it's shifting in the organic matter. And in the second one, I think what's going on is you're seeing that late in the season you're shutting down this organic matter release, and that's why in this port sample you see that there's actually more organic matter that's accumulating in the soil. And so this is just a biological way of explaining what's going on in kind of with these nutrients. I wanted to show one more of these. This is another one that I thought was quite interesting. This is looking at phosphate consumption and organic phosphate assimilation. So similar to nitrogen, again, these are the pathways that we're seeing. And one of the things that we can really see is that these are kind of basically that they're all kind of shutting down later in the season, except for in this littler one. So you can see that everything is kind of, especially in the phosphate assimilation, we tend to see a lot more of this, the yellow that shows up. We don't really see that here. And that's kind of showing up in this metric right here, where we see that we've got a high solubilization phosphorus pathways, which means it's making it more available to the plant. And that's exactly what we're seeing in the nutrient pathway as well. So the key takeaway from this project was that we were able to see some biological differences in the way that the management, basically the till versus no till, but we were also able to see some very fine scale differences between the variety of wheat that they were using. And we were able to basically show that there were differences in organic matter, nitrogen, as well as phosphate throughout those different studies. And all of this is leading to helping growers, to helping manufacturers, to helping people understand sustainability a little bit more, to understand what that means, to be able to say, okay, if I do switch to no-till, or if I switch to something that's more sustainable, more regenerative, what can I do? How can I show that? How can I see if that really is having an impact on my soil health? And so now we have this new impact measure that we have, which is this BeCrop Rate, which I spoke about a little bit, but really what it does is to me it's a little bit amazing how much it can tell you what is going on. So I've done a couple of studies where one of the treatments, because we're treatment agnostic, and so some people say, well, I want to see how my synthetic fertilizer is impacting the biology, and so we'll run that. And in some of those cases, when somebody uses a synthetic fertilizer, we can actually see between the control and this, we can see this BeCrop, basically the soil health score, it actually drops. And so within 2 weeks, we're able to see how you're affecting kind of that sustainability. It's not something where you have to wait years and years before you actually see it happening. It's very sensitive, very accurate, and it's actually quite impressive how well it works. And this is something that you can use with growers. And so if a grower says, hey, I'm trying to be more sustainable, we can say, well, let's take a BeCrop Rate sample of your soils now. Maybe we'll take it in the fall or next spring before planting, and we'll see how that sustainability index is changing. Conversely, you could say, well, let's try a bunch of different treatments, the different management practices, and let's see which ones works best. We can do a comparison and say, okay, if you're adding organic amendments, or if you're adding biological products, or if you're doing no-till or a combination of those, then let's see which one of those does the best to improve that BeCrop rate score. And really, all of this is to really empower the growers, because for too long, and really manufacturers as well, for too long as manufacturers, we have been on the side of, you'll just have to trust me. We've done all the work to show in field trials that this works. Trust me, it's going to work. And for the grower actually is able to say, well, I want to see the biology behind that. I want to see it actually work. So here's an example of some of these things that are going to show up. So like if you're doing a lot of chemical fertilizers, mechanical shifting, crop rotation, biological inputs, it kind of shows how these show up in one of these BeCrop rate tests that we've been doing. And so, it's basically is a score that falls, we can give either a score like you see here, or else we can give kind of a grade, and we're able to certify it this way and say, you really are a sustainable grower. Your BeCrop rate is high. You are using sustainable practices. And so this kind of gives some accountability behind that sustainability. It's not just about, I'm going through the motions. I'm not doing conventional till anymore, but it's actually saying, no, we want to look beyond that and say, how is it really impacting the soil health? And I think that's it. And I hope this was informative. I hope that we touched on everything that that we mentioned we would in this webinar. And I'd like to open up to any questions that might have come through. Sarah, I think you're still muted.
Sorry about that. Thank you, Dr. Purnell. That was a great presentation, lots of great takeaways, and we have lots of questions that have come in, and want to just remind everybody that a recording of the session will be provided to you within the next 24 hours. So in case you want to watch it again, it will be made available. Before we get into the Q&A, Jacob, if you don't mind, I just wanted to quickly cover the BCA program, as I see a lot of questions that have come in about the certification. I'll just quickly cover what it's about, and if you guys need more information, we'll definitely provide it in a follow-up email. As Dr. Purnell mentioned, Biomakers, we're on a mission to empower farmers, and we want to recover soil health. And we can do this now with the power of technology, obviously providing key insights to improve soil health, increase crop yield and quality, and reduce farm costs. And for several years now, BeCrop has been put to really great use on countless of projects worldwide, becoming a new standard in soil biology analysis. BeCrop is now available in 40+ countries, and we have a proprietary database of microbial profiles with more than 120+ crops and over 40,000 samples analyzed across the globe. And we're making this available to BeCrop advisors because we believe in order for us to realize our mission, we need more help and we need more boots on the ground. And this program was basically started, as I mentioned earlier, a little less than a year ago, and we have over 800 so far registered and they're certifying. And it's a free program, free education, free certification for industry professionals who want to help their growers, their clients, with the power of BeCrop. Clearly, it's a fascinating technology, requires a lot of education. The certification program, we have tried to simplify it as much as possible. It only takes a few hours to take the course online, and you can become certified. Once you are certified, you'll become an extension of our team. That means that you can provide BeCrop technology to your clients We'll show you how and how you can utilize it. You can get access to more training, marketing materials, access to our BeCrop portal where you can manage all your clients. You get access to BeCrop Project Planner to create proposals in just minutes. So it tells you how many samples you need, how many replicates you need, depending on the type of questions you have about the farm or your clients. And obviously, we have a lot of great in-person events that we'll be introducing in the new year that's open to BeCrop advisors who will get to meet, network, and as well as meet growers in their region. I mean, there's so much more I can cover. I'm sure you guys are probably hoping to get to the questions sooner rather than later. So I will stop here, but we will provide an email again following this webinar with more information about the program. And we'll also be releasing a lot of new webinars in the new year that are just dedicated to BeCrop advisors. So if you guys want to learn more about how to interpret our reports, you want to gain more access to our experts, such as Dr. Parnell here, we'll provide a lot more details about those in the new year. So we're here to support you, and we know you're here to support your farmers and your growers and the agricultural community. So hoping we can work together and recover soil health as fast as we possibly can. Without further ado, I know you guys are all waiting for our questions. What are the regenerative managements that can be evaluated? Dr. Parnell.
That is a very good question. And we have done so many different regenerative management. So anything that's going to change the biology, we can measure that. So, we've done where people have introduced grazing and different levels of grazing. We've done till versus no till. We've done different cover crops. We've done a suite of, biological versus synthetic fertilizers. Anything that's going to impact the biology, either in a hopefully positive manner or even a negative manner, we're measuring that. This is just a way of showing whatever you're doing, that impact on the biology. So this is more than just a tool for regenerative agriculture. This is a tool for soil biology, and of course that's really important in regenerative agriculture. But it's also important in conventional agriculture, and so it's not limited just to regenerative, and it will measure basically the biology up to the level of being able to tell the difference between different varieties of wheat in the field, as we were able to share today.
Amazing. A question that I think we get quite often, an important one. What temperature range is ideal for taking soil samples?
I hate to sound like the same thing, but it's the temperature that your crops are experiencing. All of this is saying these are the cycles that are going on in your soil. So if your soil is running really hot, and you say, I want to see what's going on in my soils right now, the temperature is high, take that sample when the temperature is high. If it's early in the season and you want to see what it is, we'll be able to do that. So it's, again, it depends on the question you're asking. If you're asking, what does my product do? Then as long as the temperature is the same and you're untreated and you're treated, we'll be able to tell you what your product is doing, despite whatever temperature is there in the background.
Great. Another question has come in. How do you look into functionality when working with DNA? Is this based on taxonomic information?
That is a great question. And it's really one that distinguishes us from from some competitors that are out there. So the thing that we do is we will sequence a short region of DNA, the 16S or the ITS if it's fungal. And then from that, we'll be able to say, okay, we have a list of all of the organisms that are in that soil sample. And then we'll go to a big database that we have that has all of the information about what that organism does. So for example, if we come back and we say, we know that Bacillus subtilis is in that soil sample, we don't have to actually get all of the information from that Bacillus subtilis because that's already been sequenced and we've got that elsewhere. So we don't have to spend all of that room, all of that extra money of sequencing to do everything. We just have to say, Bacillus subtilis is there, this is how much there is, and then we go to our different database and we're able to say, okay, these are all of the things that we know that Bacillus subtilis can do. And so then we're able to add that. And then we can do that with all of them. So some of our competitors, what they'll do is they'll want to sequence everything that's in there. And there are some advantages to doing that. So for example, if you said, well, I want to know if the Bacillus subtilis in my soil has a very specific, or we'll take another example of a question that we had from another group where they said, I want to know what nodulating factors there are in the soil for the rhizobium that I'm looking at. So we're not going to be able to get at that because we're not going to have that fine level of resolution, but most farmers don't care, right? Most farmers want to say, are there nitrogen fixing bacteria in my soil? And so we're able to get that high level resolution. And the benefit to ours is that you're not paying for all of that extra stuff that you don't really need or maybe want anyway. And so basically once we've done that, so I guess an easy analogy would be to say, if I told you that there are zebras and cheetahs and lions and elephants out there, I don't have to tell you how big an elephant is. I don't have to tell you what an elephant eats. I don't have to tell you where the elephant is living or that a giraffe is eating from tall trees, because you have that information stored somewhere else. I just have to say, this is what you've got out there. And that's the same thing that we're doing, is we're saying, okay, in our sample, we've got, elephants that are going to be dominant, that are going to be, eating a lot of carbon, and, we've got predators, which are the, and so basically that's kind of our approach to this. I hope that makes a little bit more sense.
I'm going to read one of my favorite questions that I think we get quite often by folks that are in manufacturing or retailers. And I think it's an important one. They're asking if BeCrop tests can be used to sample the biological product before use and provide placement recommendations on a given field.
That is a really good question. We are sampling the biology, so it depends on if. So I think there are two questions there. First is, can we look at a product and determine the biology? The answer is yes. Now the thing is, sometimes we have people that are sending us like biostimulant products that really aren't biological, and so there's not really a lot of microbes in there. So sometimes we're just sequencing the contaminants that are in there for them. So if you have a biological product and it's biological, we can do that. But there are people that can do it for a lot cheaper, to be honest. You can send it off to a sequencing facility and they can do it and they can tell you what's in there. Our strength is in all this functional stuff. And that's really where that second part of the question, and I'm sorry, Sarah's cringing because it's like, oh, you're not supposed to say, don't go somewhere else. But that second part of the question is the one that I love, because we've actually been able to do that in some small scales, because I used to work in manufacturing industry and I ran field trials. I ran, I'm used to this where it's like, this looks great. This is wonderful in my growth chambers. It's doing an awesome job in the greenhouse. It's wonderful. And we take it out to the field and it's like, okay, sometimes it works. and sometimes it just doesn't do anything, you know. And unfortunately, at that point, a lot of times it would be like, well, it doesn't work in enough places, so let's can the project and move on to the next thing. Well, the thing that we can do is we can say, well, the reason that it doesn't work is a biological answer. And so looking at the biology, we can say, you know, where does it work and why doesn't it work here? Why does it work at this other place? And like the question says, we can actually then start to get towards where we can actually recommend and say, well, it's not going to work if you've got a biology in that soil that looks like this. And so, you know, somebody could potentially take a soil sample and then we can match it back to the product and say, we've done testing and we know where, you know, this product A works and it looks like it'll work in your field. And so we can predict with higher accuracy that, you know, we're 90% sure that it's going to work in your field. And so, yes, you can apply this product and it's going to give you a return on your investment and everything is great. So it's a way of making it so that biologicals are not as kind of that trust me, it works. You know, we can get rid of some of that.
That's great. And there are two other questions that have come in, and you probably covered most of the answers for those. One in particular was the questions around if after analysis a specific microbe or two of interest that are lacking, can the farmer then be advised to apply the right type of product or what type of product to apply?
Absolutely, yeah, and that's where I hope that this technology, we're trying to get there. We need a lot more samples from growers, a lot more products tested to be able to really answer those questions, but we're getting there. What I want us to be able to do is I want, you know, a BeCrop advisor to work with the grower and say, let's sample your field. Let's sample the low yielding areas and high yielding areas. And let's look at the biological difference and then say, you know, it looks like, you know, your nitrogen pathways in those low yielding areas are low. You've got plenty of nitrogen there. It's just not being made available by the microbes. So let's put some microbes in there that can do this, that can provide that benefit. And then, you know, if we've tested enough of those products, you could say, well, you know, here's a product that will actually provide that benefit, that will open up those pathways, those nitrogen pathways or phosphorus, whatever it is. You know, we've identified kind of that, you know, that bottleneck, that nutrient bottleneck, and now we can look for a product that will open that bottleneck.
I have to apologize, Jacob. I was trying to take away your, I think you're still sharing your screen or maybe a presenter, so I stopped your webcam. Apologies. But I think it's fine now. A really interesting question, and I think, again, we may have covered it in most of the answers just now, but I think it's a good one to sort of wrap it all up. The question is about how do I use this information to design next year's program? Cover crops, cash crops, applied biology, fertilizer application, etc., and improve my soil biology?
Yeah, that is a good question. So one thing is that you know, we mentioned the BeCrop Rate. We've also got some other metrics that are not nutrient type of metrics, soil health, soil quality type of scores. And so one is looking at some of those, even the nutrient, you know, unlocking some of those early in the season, because it's not saying that you are, when we show that like nutrient pathways are high, we're not saying that you are burning through those nutrients. We're saying that microbes are cycling those nutrients actively. And you want microbes to cycle those nutrients actively, because then it makes that pool more available to the plant. And so even if you take it in the off season, if you take a sample and say, well, you know, my nitrogen pathways are low right now, but I don't know what they're going to be like later. If you can get those nitrogen pathways high even early on, then it's going to provide that benefit later. So it's not like, it's not like traditional nutrients where it's like, well, they're high now. You know, if you apply it at the wrong time, then it's all just going to leach out and you won't have any nitrogen later. It's not really the same when you look at these kind of biological pathways and cycling.
Maybe we can take one or two more questions. There are tons more. So again, we apologize if we don't get to all of them. We will definitely follow up with an email. We have a question from Marina. She says, thank you for an interesting talk. And she's asking, considering the shift within the community, at what level did you need to go to see the differences you shared in the case study? At the level of the genera, I think is what it's asked here, if I understand correctly.
So yeah, that is a really good question, and it's one that I don't think I covered very well, because you know we can look at the taxonomy, and I mentioned how we look at the taxonomy and how we can look and say, okay, well, here are, you know, elephants and zebras and gazelle, you know, whatever. But then at the same time, there are functional groups that are very specific to certain microbes, and then there are broad range functional groups as well. And so when we look at functional groups, we don't look at the taxonomy. Instead, we're looking, like, you know, if we looked at those categories of the elephant, and I don't remember what, but if we said herbivores, then we would say, well, you know, elephants aren't related to gazelles, and they're not related to giraffes, but they're all herbivores. So that functional category is the same, even though they're very different taxonomically. And so when we look at that functional level, we're looking across all different, and sometimes they're very different taxonomic groups. And so that's really, because there are a lot of groups that can look at just the taxonomy and tell you who is there, but where it gets really difficult is telling you that function, tying it to that function of telling you what they're doing, and being able to give you those nutrient pathways and, you know, the plant interaction pathways like phytohormones and stress adaptation pathways.
That makes sense. There are several different questions that have come in about the test and where to purchase, costs associated. I have sent a link in the chat here, and you guys can review that and check out the page for the BeCrop test, or visit our website to learn more about BeCrop Technology. Again, the BeCrop advisory program, there's a link on our website that you can visit to learn more, and we'll provide obviously more information on a follow-up email. Well, Dr. Parnell, I want to thank you for your time and sharing your knowledge with us. We have a lot more webinars with Dr. Parnell presented in the past that are available on our website, and there are more coming in the new year. So please stay tuned, check out or be on the lookout for our emails. And if you have any questions or thoughts or want us to present on in a particular topic, please reach out to us and let us know. We want to hear from you, and we want to be able to create content that's useful for you guys, and want to make sure we bring that all to you either through a webinar, blogs, articles, or ebooks. Dr. Parnell, thank you again for your time. Thank you everyone for joining. We hope you have a wonderful rest of your day, and happy holidays from Biome Makers team to you, to your farmers, and to your clients and the rest of your team. So thank you for joining us today.
Thanks, everyone.
Thank you. Bye for now.